Hello, it is really my pleasure to introduce Heng to GIS community. So I remember my first talk at GIS seminar, and I mentioned that I have 3 role models in my work, and Heng was among 3. 2 others was Esko Conan and Genome. So I think that most of you know about Heng. So he actually did his PhD in biophysics, and but he worked at that time in genomics. It was just about title, and he worked shortly with BGI. Then he moved and did his postdoc with Richard Durbin, also one of giants in the field, and then moved to US. Broad Institute, and now he's a social professor at Harvard Medical School and Dana-Farber Cancer Institute. So I think there are so many things that Heng did in the field, including SEM tools, VWA, Minimap tool, some HiFIAS, and so many things. And now he really helps to move forward in this pan-genome field, and I'm really happy. Thank you.
Thank you. That's all for the introduction, and it's my great pleasure to present my work here. And so I might actually have another presentation tomorrow. So this talk will focus on the long-read sequencing, and the tomorrow one will be focusing on this the pan-genome. Okay, it's okay. Firstly, this is actually adapted from another slide deck, and basically this what we are doing is basically it's more all the modern research based on sequencing. This is kind of based on our tools. In particular, you may see the other version is actually showing this descent. No, we are not this descent. We are not that fragile. It's we are this descent. And so we have the short read, we have the SAM-BAM format, and DW alignment for long read, Minimax alignment, I have SM assembly. And for all of these tools are widely used in the field. And in today's talk, I will touch upon 3 topics. The first is the long read sequencing technologies in general. And so the second is the long read assembly, and actually many assemblies, not its applications. And then the third part is the long read variant calling. I will start with this long read sequencing technologies. And so there's in this field mainly, this is more on the background. And actually for this audience, possibly many of you have already known this, but still, the detail about 2 companies. There are 2 companies are doing this long read sequencing. The first is the Pacific Biosciences, this is in US, and then Oxford Nanopore Technologies at Oxford in UK. And so we usually say the ONT or Nanopore. And both of them are doing single molecule sequencing. If you have enough DNA, you don't need the amplifications, and you can see the base modifications and so on this thing. And there's a BGI has recently have the cyclone-seq in 2024, but I would say this still doing the novel sequencing, but still not quite there yet in comparison to the Nanopore. The Nanopore had been there for like many years. And so I'm not going to talk about the cyclone-seq because I personally haven't seen the data, so I don't know what the data quality. I thought the history about the 2 companies. PacBio, the theory, they have the first paper about how to do sequencing using this technology. That was in 2003, and Nanopore is 1996. It's actually much earlier than PacBio 3. But the 2 companies are founded around the same time, 2004, 2005. And PacBio has the data first. This RS, that's the first machine, 2011. And Nanopore a bit later, the first machine is Million. And so the PacBio, and then the PacBio have this high accurate rates, that's called high-fi rates, or that by high accurate rate, I typically mean this the above 99%, the average about 99% base accuracy. And the Nanopore is a bit later than that. And why I separate out this accurate rates? This is in 2022. This Nature Methods, they named the long read sequencing as as the method of the year. And that's mainly driven by the HiFi. Although it's says still long race, but at that time, it's mainly driven by this HiFi race and also the Oaklawn race. And if you look at the PayPal stock price, I mean, there's a huge jump from I think $5 per share jump to $50 per share in a couple of years. And this mainly owing to this new technology. And there's a different instruments doing this sequencing. I will focus on the high throughput sequencers. By high throughput, typically, I mean, you can get one human genome sequence to 34 coverage in one run. And so we have the PacBio review, this called PacBio, and also the nanopore, this PermDown 24. 24 means there are 24 pores and nanopores. And so you can sequence from each one. And the nanopore also recently have this P2 solo, the visual computer and P2 integrated, but they have 2 nanopores on this device. At smaller scale, there's these others, it's more like benchtop or even just handhold, like Million and Blanc, you can put it on at your hand. But here shows this the instrument, the high throughput instrument. You basically can sequence the human genome in a few days. The PacBio is actually of height of a human person. I mean, Google this picture and they just show the right side. And this PermDown, I think this is 24. Actually, I don't know what's the size, but it's much smaller than the PacBio. And then this is the early access product. I'll actually use the fake laser here. This is the P2. They have 2 nanopores in here. You can see this is much smaller than both of the machines. And in terms of the price, the instrument costs, the review and the nanopore, I'm showing the P24. I got this price from their website. And the review, each time they can do 4 flow cells. And nanopore, the P2, the 2 flow cells, with 2 nanopore, 2 flow cells. And P24, this is 24 flow cells. And this is early access product, so it's not formally ready to the market. And so the P24 is earlier. And so the instrument cost, the PacBio is over $500,000. And so the P24, there's almost $500,000 as well. But the P2, actually, I really like the P2. I actually don't know why they have still put it at early access stage. And usually that means that it's still not mature enough. But I don't know what's the issue there. But you can see it's very cheap, and there are 2 numbers. One is with the onboard computing, and the other is without onboard computing. Without onboard computing, you have to put it connected to your desktop or to a desktop to do the base calling. And with onboard computing, it comes with GPU, and so you can run it. And the laser chemistry, okay. And typically the per run, and remember that each run you can get 4 flow cells with PacBio, and with P2 you get 2 flow cells, and each run takes about a few days, 24 to 1 to 3 days, and also depending on the amount of the library. And each flow cell gives around 30 to 44 human genome. This means that if you do a run once, each flow cell gives you enough data for the assembly for the very calling. And the reagent costs, this latest reagent costs, again, got from the website. It doesn't include the human cost, doesn't include the instrument cost, doesn't include, say, electricity and all the other things, just the chemistry. And the chemistry, the claim is $500,000, $500 for PacBio. And nanopore is roughly around the same range. And in comparison, even NovaSeq X, they claim $200 per genome. And so they can do, but the throughput is much higher. They can do 64 genomes per day. And this is, if you look at these genomes, the nanopore PacBio, they can only do, say, 4 to a few genomes per day. This is, the throughput is like more than 10 times higher with the Illumina machine. This is actually important if you want to do this population scale sequencing. And with PacBio, you can just count. Each year you can only sequence a few several thousand genomes with one machine. But the NovaSeq X is the throughput is much higher. And so there's the data and data quality wise, the PacBio is a bit shorter, 10, 20 kb-ish around that range. And nanopore depend on protocol and also the library construction, they can reach much longer. The longest nanopore range is several megabases, but it's very rare. And so people call this a whale watching. Basically, they run it and until they see a very long range, several megabases. This is just like how you watch the whale on the sea. It's very, very advanced. And but the point that they can reach that length. In terms of accuracy, PacBio is better than the nanopore, and the PacBio is actually on par or sometimes more accurate than short rate. I mean, say short rate here, it's actually could be given companies, they have different accuracy. Some companies, short rate companies, they more recent ones, they can reach very high accuracy. Actually, they are better than nanopore, better than PacBio. But I mean, the PacBio is better than generally is better than Illumina, except in the homopolymer. This systematic error. Okay, I couldn't find Google a figure. I haven't seen that figure, but I couldn't find a paper. On the left, it shows the x-axis, this shows the read accuracy, the QV, and this 30 means 1 per 1,000 error rate, 40 means 1 per 10,000 error rate. On the y-axis, this is the read length, and just to look at this PacBio, and so the read length is about the 30. Okay, this one is about 20 kb. And the error rate is about 30 QV30, the 1 per 1,000 error rate. Actually, the mode, the peak here is better than that. And the nanopore is showing the duplex data, but for simplex data, and so the read length is about similar 30-ish, and you can go as high as 50 with simplex data. But accuracy with simplex data, it will be around 20. Here is the duplex is doing the double-strand sequencing, and so the error higher. However, this probably nanopore duplex, I've heard that it had been already discontinued, but heard from others. This is the figure from this Sergey's paper. Okay, between the PacBio and nanopore, and so the two companies, still back to the two companies, and PacBio generally is more stable. And they develop products in-house and you don't know what they are developing. But when they announce it, it's production ready. You can directly use that and put it in production. And nanopore, I call it innovative. They have very exciting early access products, including this P2 Solo, the PerpSion 2 machines. But some succeeded, some and other field like the duplex, that is discontinued it. And a lot of their products, they have discontinued quite a few products. I mean, but the R10 is a successful one. And this is the dominant technology at the nanopore. Okay, this is exciting about these sequencing machines, but they have both actually all along basically both companies have somewhat severe limitations. And I have shown the throughput is much lower. It's 20 times or even more lower than say NovaSeq and like Ultima, and also the BGIS, the biggest merchant. Actually, don't know what's the name, but it's the throughput is much lower than those. And so when we do population sequencing, this will become an issue. The second problem is they require a lot of DNA as the input. And the PacBio, the latest chemistry, they claim they can do, have a mammogram. But the nanopore, the standard simplex sequencing, it requires about that. But if you want to do ultra-long sequencing, you need a lot more DNA. And typically, you can only get this amount of DNA from cell line. It's difficult to draw the blood to get all of this DNA. This has become a severe limitation. And also, the ultra-long, the quality varies a lot. And I actually see other similar, the nanopore and the PacBio depend on the tissues. For some tissue, especially cell lines, you get the best quality from the cell lines, but the quality become lower if you draw blood. And if you do this control, you prepare the libraries using the tissues, say from the livers, from brains, this will become a lot more difficult. And so the read line typically become a lot shorter and throughput also reduced. And this DNA has been considered problem. They are improving, but not quite there yet. The short rate is a lot lower, like one even in the case, some cases it is 2 orders of magnitude lower than the PacBio and the And so this the DNA basically also makes it difficult for the clinical applications. If you want to do tumor sequencing, you typically get a little bit about the tumor tissues, and if you do the sequencing, actually they typically don't have enough DNA to do the long-range sequencing. And then this also they like to high quality tools, and we have this day we don't have the GTK for long reads. We are doing quite well on the assembly, but on the variant calling in the third part, we'll see it's actually not quite there yet. And also there's killer biological application at scale. So far we don't know yet. There are a lot of the people biological we can study with the short reads. They have already solved a lot of problems. And so we do know that long reads can deliver, give the additional information in the complex retrietions. But actually, that the question is it worth it? It's a lot more expensive. And the ultimate genome is they claim $50 per 34 genome. And the long reads is 10 times than that. And so it's 10 times more and more worth this all these things. And so far we don't know. Anyway, but still, I mean, this it's actually also the both companies are struggling. It's financially. But I would say this in the research is actually still you use a lot more. You can see this is the citation of the minimap tool over the years, and it's increasing. Basically, these days, if you do long read analysis, I mean, most of them will use almost everyone use the minimap tool. And you can see this is still increasing because the scientists are using this long reads, and the trend will continue. But then they are struggling mostly because they don't sell the more machines. And for them, if you already have the payable machine, you don't buy the machines again. And so that's their problem. But anyway, they hope this both company stays and they are continuing with each other, and this is good for all of us. Yeah. Okay, this concludes the first section. This is long. We have the long read first generation long read are from, but these days are very accurate, even better than the typical 11 rephrase. Reagent cost is higher, but it's not a lot higher than some other technology. And their limitations is low throughput, high data, and it's gaining popularity in research, albeit the financial difficulties of both companies. Okay, the second part is the long read assembly, and the first is why we need the assembly. And I will start with short read. This is from this Tagwell paper in 2019, the Hi-Fi paper in 2019. And this is on the x-axis, it's showing the accuracy of the structural variant calculation, mostly the long insertion and long deletions. And you see this, the x-axis shows the length, the y-axis shows the accuracy, and the orange curve shows the sensitivity, and the blue curve shows the precision. And this shows actually this shows the long reads, not short reads, sorry. Why I put this here? Okay, anyway, this is the long reads variant calling accuracy. This is you align the long reads against the reference genome, and you do the variant calling. And you can see there, accuracy, especially sensitivity, falls off around 5,000 and 7,000 base pair. Both of mostly the precision, and they become a lot lower. Okay, why this is swapped? Okay, and so you can see this Illumina. This accuracy is a lot lower, and then this okay. Anyway, just focus on this. Okay. Anyway, this you can see from this long read alignment. The accuracy will drop at the longer scale, and that's mainly because once you have events longer than the reads, it will be very difficult to do the calling from the long reads. And with the assembly, you can build the assembly through the longer reads, and so you can go through the longer regions, and so this will improve the variant calling. And so this shows the narratives, and this is actually screenshot. And on the top, these lines each line is a long read assembly. At the bottom, this shows the read alignment, Hi-Fi read. This is Hi-Fi read alignment. You can see there's a gap here. There's no read map here. But if you do the assembly, you can actually see there's here that event here. The parental habitat are different. And so what's happening here is that this gene, the GTF2LD2 gene, and they have a very close homologs downstream to this gene. And then for this particular sample, and there's a gene conversion, the non-allelic gene conversion. The typical gene conversion happens between habitats, and in this case, the gene conversion happens between the two copies of the gene. And so this gene, this part overrides the region in the middle. As a result, this read region is because the read coming from the blue part will be similar to this. As if you do the read alignment, and so the read coming from here will be mapped over there to the other gene. And this is called the false read alignment. And so this is why you see this homozygous event. And so that's why you see this there's a long gap. But with the assembly, these two copies are not identical. These are not identical, and the assemblers can just see this few bit difference. They can separate the two copies and place them back to the right place. And when you do align the assembly, then you start to see all the signals. And this is the why the assembly is important in this case. There are more complex cases like this. Okay, this is typical because you can see this region is longer than the rates. But everything longer than the rates will be challenging for the alignment-based methods. And this is a second example. This is a C4 gene. There's a segmental duplication, but if you align the genes, you can see here there's long gap, the CKB long gap, and you can see the coverage increased. As you know, there's copy number changes, but you don't know what's really happening. And basically, what's the linear structure of the gene? If you only look at this alignment, you read the alignment, you don't know. And what really happens is that the maternal has one actual C4B genes. There are C4A and C4B that they are homologous genes. They have actual ones, and so in the human population, the C4 gene look like this. Most of people have one C4A, C4B, and this includes the SAM13 and also the GIS38. They have this most common habitat. But in other samples, for this sample, it's actually come from come from this habitat. This sample has one C4A, but mother has two C4B genes. And so the other copy will fall on top of this. And so we created this. With alignment-based approach, you can detect this actual copy, but it's very difficult for to tell whether this is the actual copy is C4A or C4B. You will need a lot of more effort to make the inference. But with the assembly, you can jump through, you can easily resolve this problem. And then also not only the two genes, there are also other genes like these genes, and also they can produce similar issues. This is quite pervasive in the human assembly. And on the biological effect, this plot shows that if you have a higher covenant number, typically you have this higher expression, and so they have a biological effect to that. And so back to the assembly, how do we solve this? Do the assembly. Very basic primer about the assembly. Typically, when you do the sequencing, shotgun sequencing, the black box is the genome. Initially, we don't know. And so in the wet lab, and because the current sequencer cannot sequence the whole chromosomes, the human chromosome line is 250 megabase. The current sequencer cannot sequence them. And so they have to chop this genome into small pieces and then amplify them. And sometimes we don't, and then we do sequencing. And for each of them, they call the rates. And then we observe the similarity between them. We can reorganize them. They're the same rates, several rates. But if you reorganize them, you can see these parts are similar. And so you can, based on the similarity, you can recover this the reference genome. And in this field, and people also take the assembly as analogous to the jigsaw puzzle. It's a very close analogy, actually. Now here is the read piece. Each piece is a puzzle piece, and the whole picture is the genome. And how to resolve the jigsaw puzzle? Basically, you observe the similarity between the two reads. You either see the niche or you compare the picture, and so you piece them together. This is like the overlap assembler. And so when we have them, and so you can reproduce the picture. Of course, in practice, it's more complex than that. The real data is closer to this, actually more complex than this. First, there are similar patterns. It's repeats in the human genome, similar duplications, and like the similar genes, and also the sex telomeres, and repeats. And then we have not only have 15 pieces, we have 5 to 50 million pieces. These are the rates. And so there are maybe missing pieces because of there's coverage drops, there's a signal bias. And so they may call even coverage, you may miss some regions. And most pieces are damaged. It's rare to see a perfect rates. And so you have to do the error correction and do all these things. And so they make it difficult. And the most challenging part is actually, if you take this as analogy to the jigsaw puzzle, but it's not quite right. If you do the jigsaw puzzle, you have the picture. You look at the picture and peel them back. In some way, it's a mixture of the overlapping and also the read alignment. You are doing the eye alignments, align this piece die to the picture. But when you do the assembly, you don't have the picture at all. Just imagine where you're doing jigsaw without looking at picture at all. You don't know the whole picture. You just have a couple of pieces and you want to reconstruct it. And so this is more challenging than the jigsaw puzzle. And that's why it's the assembly is actually has almost 40 years of history. It's one of the oldest and one of the most important problem in the field of computational biology. And it's still it's ongoing. It's because of all these challenges. And the assembly, although the assembly has more than 40 years of history, but it has rapidly evolved in the past 4 years. And if you check the assembly like say 4 to 5 years ago, these are actually it's crappy in comparison to one assembly. And what's the difference? In old days, human deployed, you have a paternal copy and maternal copy. Typically, several years ago, people will crack them. For default genome, you only get one assembly, and it's a bit removing all facing information. And it's also most copy genes are missed, and all this problem. But this is solved by the recent assembly. And this plot shows, what had been changed. And in the plot, each on the axis it shows different assemblers. On the leftmost part is two. Recent assembler HiPySM and HiPyNode optimized for the recent data, and all the other is older assembly. And the color shows actually shows the HiPy data. This is null data. I think this CRR is older type of data. On the y-axis, it shows the error rate, and I'm not going to depend too much about what it means. But basically, the error rate, the lower the data, better. If you have 0, means you make no mistake around these duplications. And you can see for all these older ones, sometimes they have a 70% error rate. And even the better ones have 20 to 30% error rate in one copy genes, which means they will lose. If the gene has multiple copies, they will lose these copies in the assembly. But if you look at these more recent assemblers, they achieve below 1% error rate. They rarely miss these amount of copy genes. And there is a difference between, say, 70% versus 0.3%. This is actually 2 orders of magnitude improvement over just around 2020. And these days, is we all routinely achieve this and even better than this one. But this is mainly driven by the accurate long rate because of the data. And how do we do the assembly? I will go through this process, how we do the assembly. They show the old number data. This is not the latest one. I will show the latest one later. This old number data, you can see this is this gaps and the color bar, these are all errors. And you can barely see what are the real. This might be real SNPs, but what about this one is the real SNPs? It's if you only look at this, it's very difficult to tell. But with the HiPy data, the way the HiPy data changed the game. You can see these very clearly. These are these kilo-level SNPs, and these are well faced. You can see here there's a multiple. This really doesn't have this color bar, doesn't have the kilo-level SNPs, but this one has it. It's clearly faced. You can use this with this information, you can do the error correction. With this, with help, the old number data very difficult to do it. Even human eye can't tell. But with the latest the number, the HiPy data, it becomes much easier. You can decide the remaining errors. You can correct away almost all their errors. Over 99% error, you can correct away over 99% errors. You get very clean data. And so why we need this clean data? Why we want to see this? This shows the example of how to do the deploy assembly. And so here, there's a paternal maternal half. This shows the heliodiodes, and these are the rates. And so if you want to do the assembly, we typically we do this overlap assembly graph. And so each rate is a rate 1 corresponds to rate 1, and so the rate 2, and we add arrow between them if there's an overlap. And so we can see between 2 to 3, the 2 to 3 there's overlap. And so there's an edge between 2 to 3, 3 to 4, there's an edge. And so what's the dotted line? Dotted line is the terminology they call trinity edges. In some way, it's sort of redundant edges. And if you look at this, the 2 and 3 have overlap, 3 and 4 have overlap. Based on the overlap length, we can infer there's an overlap between 2 and 4. Basically, we don't need to see this the overlap between 2 and 4, but with 2 and 3 and 3 and 4, we infer the edge between 2 and 4. And so this is a kind of redundant. And so these are similarly out dotted edges are redundant. Because they are sort of redundant, we remove them, then we get the assembly. The problem is the input is 2 genomes, but we end up with 1 genome. And we can see this red and this blue alleles are mixed together in this. This is old assembler will have this. And default genome and single genome out. And what this question, this high accuracy rate make possible is that you can, from the, because it's near perfect, you can see here that there was alleles on this ridge. And then, and for example, this 4 and 6, with all the methods say 4 and 5. With all the methods, you would collect them because that means you can't really see the difference. I'm not accurately enough. With the latest assembler, we can see the 4 and 5. There's 1 base pair difference. But even this 1 base pair difference, that's enough to separate the two rates. And so you will not have this 4 and 5. You will not have the overlap between the habitats. And similar 6 to 7. And so when you remove the trending edges, you will end up with this graph. This graph preserves the heterozygotes and as well as the phasing. There are 2 SNPs, but this keep the phasing between them. This is the key difference between the data and all the assemblers. And so, I mean, we have this, I mean, putting them together, we have this, they have a race, and we have the assembly graph, and so this is unity. But this is not upon, this is not done yet. Okay, this shows the real data. There's around 2 megabytes in the human genome. And so we usually call it bubbles. You can see the long region, these are phased. And so you can, so you have this, actually this just extension to this, the real data. And when we have the parent information, we can tag this unity based this sequence here. This is about, I guess, 50 kb-ish, maybe a few hundred kb-ish region. We can tag it with the parental information. And so this is already tagged. And so we just trace the red line. This is the maternal habitat. You get the maternal quantity in this region. We trace the blue line. We get the paternal quantity in this region. And so this is how we do the assembly with patient. And the problem is that often we don't have the parents information. And so what do we do? And so we can further to improve this graph. And so this basically this graph is shows that is correspond to this graph with the multiple bubbles. We have multiple bubbles. And so here we can use the outcome rates. They are much longer rates than the high rates. And often the outcome rates may span multiple bubbles. In this case, these thin lines, these are the outcome rates. And so we can use this information to further separate the region. For this region, with outcome rate, we can completely separate them. We can separate the into 200 without the parental information. And another important data type is the high C. Because even if with the outline, we get longer bubbles, but the bubble, we call the bubbles. But for the with the outline data, the bubbles is still, I mean, maybe 10 megabytes, but still not the whole chromosome. The whole chromosome is 250 megabytes. But to resolve this whole chromosome, we need new data. It's called the high C. I'm not going to the details of the high C. This basically is can further separate the 2 habitats and across the whole chromosome, reliably face the whole chromosome. And currently, the high C is widely used for some new species. And if you want to get chromosome assembly, high C is a required data type in addition to long rates. And overall, the modern assembly workflow look like this. We start with the raw rates, and then we do the error correction. I mentioned that you can correct most of the errors away. Then you generate the assembly graph, and then you add outline to further simplify the assembly graph from the full rectangle. And you cannot resolve with the shorter rates, but if the outline rates, you can resolve the red topology. And you add the long range data high C to further do it. And this is done through multiple papers in my lab. But in the past 5 years, I would say the major contribution of my lab, the most important contribution of my lab in the past 5 years is the assembly. This is done through, although I'm mentioning it's just like 10 minutes, but this is a long, 5 years of hard work. I'm doing this. And the on left shows the homozygous genome. And here, heterozygous genome is similar and still following the same procedure. The main difference that the graph already look different. And so the heuristic may be different, but the theory is about the same. The modern assemblies, we have the first result of human genomes, and we can routinely achieve the T2T assembly. Typically, we can resolve a third. I think the more recent as 2/3 of the chromosome to T2T we see in our data. And we can routinely do this for the non-human species. This has been used a lot. Post-assembler have been used a lot on thousands of such species. Okay, so far I'm more focused on the high-fidelity assembly. And so what about nanopore? I only mentioned nanopore ultra, but what about doing the nanopore only assembly? We only have nanopore data. And so the older nanopore rates are too noisy to assemble. This has can achieve some, but still difficult if you use the older data. The quality will be lower than the high-fidelity assembly. But the latest nanopore rates is a lot more accurate. These are achieved 99%, but still not good enough for most of the high-fidelity assemblers. And Hero can do this with the deep learning. And one potential issue is the computing intensive. And so very recently, just I think earlier this year, and so high-fidelity now support the nanopore only assembly. They can take the simple rates only and to do the assembly. Here's some basic results. They're still around the same region. And so I showed earlier, the nanopore rate, the old nanopore data. There's a lot of noises. And this is the latest nanopore data. And you can still see this, the purple things, these small insertions. This you can see there's still error, quite a lot of error, but this is way cleaner than the data we got several years ago. And with this, because it's easier to do the error correction, we can error correct most of the rates. There are some, you can see these rates, and also some rates are not corrected. If they have a higher symptom error rate, some may be difficult to correct. But in practice, this rate will be ignored during the assembly. And so the assembler will use this rate. If you look at the other rates, there are almost no errors on this rate. And basically, with this error correction, we can just apply the same procedure I showed before to do the assembly. And the following steps are similar. You can still use alpha-long rates, you can still use high rates, you use the way to separate, and all the following steps are similar. And here shows some using the H02, the rates. And so this is a different nanopore run. At the bottom, this is a high-fi run. And you can see the main advantage here is the counting N50. The length is actually a lot longer than high-fidelity. High-fidelity is N50 about 50 megabits, 60 megabits, but with the nanopore, actually the coverage is a bit higher. Even at the similar coverage, this is better than high-fidelity. The QV, here it shows the two different the base color counting mode. Super accuracy, the most accurate mode of the base color. This is the medium accuracy base color. And you see the QV, the considered quality, there's a 3-point difference, which means about 2-fold the difference in the base accuracy. High-fi is still better than the nanopore, but the nanopore is not far behind. And most of the error come from the home polymers. Beyond the home polymer is actually the difference not that big. And this shows more the data sets. Here, this blue dot shows the nanopore data, and this orange, this high-fidelity data. The x-axis shows the read depth of the data sets, and the y-axis shows the contiguity, the counting N50. The longer, the better. And you see at low coverage, this actually both neither of them around 30-fold, neither of them doing very well. The high-fi is actually better than nanopore. My guess is that is because the go back to here. Because the discard rate, it actually have to discard a small fraction of the data. And so at the low coverage, and we lack the power to do good correction. And that's why at low coverage, actually the N50 not that good, only around 20 megabits. But once the coverage reaches 40 or something, you can see this is start to doing better than the high-fi. The high-fi is plateaued around 80 megabits. This is limited by its the battery length. And because the nanopore rates are longer, typically 30 to 50 kb if you are doing the library well. And so you can see that this reach a much longer N50. this is actually in terms of the connectivity, non-core is better than HiFi. Don't have enough coverage. And there's also we compare the HiFi versus the Workle and the plus Hero assembly. I think generally this N50 is comparable. This is HiFi this is Workle. Maybe a bit longer, but it's around the same range. And the QV the Hero is doing better in terms of the QV, a little bit better. Not like twofold, but it did better. In terms of runtime, I maybe have better runtime, mostly because this is and also we don't need the GPU. This all CPU only need model CPUs, and you can run this. And it's more streamlined, just one line. You do the error correction and all the assembly through one line. And also this okay, this is also the HiFi okay, this is non-human data. This is fish data, and so this is the Hero. You can also combine Hero and HiFi versus the network-only mode. And the accuracy is, I mean, the N50 roughly about the same, but at least on the data set, the HiFi the ONT mode consistently gives better, longer context than the Hero plus the HiFi mode. And yeah, generally, I would say this the Hero and the HiFi ONT is roughly comparable, and so the Hero is better at QA. HiFi sometimes better. I say a little bit better in terms of N50, but really the difference is small. They are comparable. And HiFi mainly is better at is far faster. And in terms of the ONT assembly, it's now the days we can do not only assembly, and so it's achieve better connectivity and higher coverage. Mainly we call the longer rates, and it's even more even coverage. And so I don't have data to show here, but in birds have this called microchromosomes. The bird for the chicken, they have I think 38 chromosomes. I think a third of them, these are the microchromosomes. These are short chromosomes. They sort of like human eccentric arms. They are I think only a couple of megabytes in length, but they are highly repetitive. And the people are having problems to assemble these microchromosomes. If you look at the HiFi coverage, there's a huge drop on microchromosomes. And so, but Nanopore doesn't have this problem. It's actually Nanopore in some way, and Nanopore kind of doing a lot better than HiFi on this microchromosomes in bird. We have seen multiple data sets. I think that this is real. And the ONT has higher QV mostly in polymers. And the future, I think the future is if you really want to get higher quality assembly, it will combine the Nanopore data and HiFi. So the HiFi has better base accuracy, and Nanopore has a longer N50. If you combine them, supposedly you will be doing better. Currently, we don't have a assembler that at least HiFi cannot really do this. And with Workle, you can correct this Hero and then combine with HiFi data, but that's still treating them as the same data type. And so this is still not the best solution, but there should be better solution in future with combining the assembly. Okay, this is about okay. Actually, I haven't finished about the assembly yet. Tumor assembly. We are trying to do the tumor assembly, and it's really messy. We here guys start something here, and so here it shows the sometimes we call it this digital cell type. This look like the cell type. You can see this is one chromosome. Okay, I should use this laser, and this is one chromosome, and so there's a translocation. This the coordinates. This is the translocation point between the two. I mean, on the arm, and similar here. And you can see the complex ones here. This in this region, this involves the small segment 10q. Involve multiple small components. And for this one, for this junction, it shows here. It's actually, and then for this, it corresponds to here. You can see there's a lot of the detail changes here. And also, for example, for this region, it shows here. It's actually going through one segment and then going to reverse complement of another, the segment of another chromosome, and then to a third chromosome, and then goes back to the second chromosome, and then goes into the first chromosome. And there's the direction changes strand changes from one strand going to the other. This we usually call the fold back. And we can see, But all this, I mean, it's done, it's semi-manually. There's lots of manual work when creating this. And also remember, this is cell line. They did a correlative line. They did cell line data. If you do parameter tumor, there will be more subnormal events. And I don't know how to resolve that. And even for this cell line is a represent simple cell line. And so there are not so many events. And if you look at some more, let's say breast cancer and so on, some of they have this generally is a triple S. And you will see HCC 1954, there's lots more, there's thousands of structural variants. I mean, the translocations event. Here, there's only like 50 to 100 events. But in those genomes, they have thousands and tenfold higher than this. And we don't want to do that manually. And so basically, so far, we can resolve a lot of simple ones, and it's useful, but it's labor intensive. And I'm not quite sure how to do that automatically. So this is a very inspiration. And in addition to cancer assembly, I mean, they are doing this metagenome assembly. And in our paper in 2022, in this one paper, we get more circular, it's called metagenome assembled genomes. We got more circular genome than all the circular genome published in the entire literature at that time. This just shows how much we can do with assembly. And these data are even better than not from us, but from others, and achieving the same. And in addition to the HiFi, these are the only work for HiFi data. And so the developer has left. She had joined the ONT. She developed a metagenome assembly with HiFi, but she moved to ONT. And she no longer doing that. But Jim Shaw, new postdoc in my group, he is still doing this non-core assembly using the simple data only. It's currently temporary name, the MeloSM, it will change. And basically, you can see it here, you can get circular genomes. And you can also, I think this is also in here. This is in comparison to, I think the MetaFly or MetaMDBG, I forgot. I think the MetaMDBG, it gets one context, but it actually is MeloSM, separate them into two. Okay, I think this is the MeloSM assembly. There are three genomes in this genome. And so two of them assembly into circular, and the third one is assembly into this. And they can separate very similar strands, and the other assembly don't work well. Okay, this is a real summary. And so the summary that there's a 98-day difference between the old assembly and old assembly. And with one data type, there's either just one HiFi data type or one simple data type. Even one data type, we get better assembly than GR38. And GR38 is 20 years of effort going to, more than 20 years of effort going to this assembly. But this, I mean, we can do that in 1 day and with one data type. And with three data types, HiFi or simple nanopore and ultra-long, nanopore and high-C data or parental data, we can assemble typically multiple chromosomes into T2Ts and 1/3 to 2/3 each. This is much better than this all the current, most of the current genomes. And older assemblies, I would say, are irrelevant. If you find the older assembly, human, non-human, they are much worse than if you want to do study, I'm going to focus on new ones. And nanopore assembly is on the rise. I think there's still a lot of things to do, but it's now competing with the HiFi assembly, at least in terms of the connectivity and also the management assembly. Okay, this the third part, and yeah, guys will be a bit brief on the third part, which I focus on fusing. Okay, I showed, I mean, in the second part, I showed example why this nanopore, why the assembly is preferred over the reference-based alignment, reference-based variant calling. And so this actually the opposite reason why this reference-based variant calling, we still need reference-based variant calling. The first is when we do the assembly, we still want to find the SNP location on the reference genome. And so we need to do the assembly to the reference alignment to find the variants. And secondly, the assembly, because it's a more complex process, and it's actually sometimes may miss small variants. The accuracy is lower on the small variant. It's better on the large scale, the very long insertion, longer than the race, but actually accuracy, the base accuracy is lower than do the SNP calling. And assembly is computationally intensive. It's lots lot more efforts than doing the read alignment and reference calling. And lastly, if you want to call semantic events, the assembly will remove a lot of semantic events. And so if you want to call semantic event, you have to do this alignment base. There's about 5 events, about 2 or 3 reads. It's very likely the assembly will just remove the reads, taking them as significant errors or rare events, and need this alignment to do the semantic. Okay, there's actually, this is not a new problem. There's already been a lot of long-range variant callers. I'm doing the genome lines and the semantic structural variants and small variants, and there's also the diffusion tool. So why we need a new one is the problem is that we have lots of them, but what's missing is the integration. And you think about people are doing this because it's in short read world, and these are separate. They are separate because you need a separate algorithm to tackle each class of for the small variants and trivial variants. You need a very different algorithm to find them. And also the phasing. Short read doesn't have the phasing. And so you need the actual thing, the other things to the phasing. But the DE is probably separated. They are not connected. But in the long read, it's actually different. If you think of SNPs and SVs, these actually essentially they are the same. They are all, if they are contained in long read, these are the same events. It doesn't matter whether it's your in-out base pair or Mason base pair. You can be resolved the same way, and they can be phased together. And phasing actually delivers a lot of powers. And so if we do like the old days, short read, if we separate them, they actually lose power. And this is why, okay, this shows a example what I mean. And so in this habitat, there are 2 habitats. One has a 16 KB insertion, the other has 11 KB insertion. And this group of reads have the 16 KB, this group of reads have the 11 KB. You can see there's a heterozygote here. They just separate the 2 habitats. With a single SNP, you can, here actually there are also another heterozygote, but based on this SNP, you can separate the 2 habitats, you can do spacing. The SNP is quite clear. But what about this? All this clipping, this caused by this 16 KB is longer than the reads. And so it's very rare for a read to get the correct alignment. Only 2 reads out of I think maybe 30-fold, 40-fold reads get the correct alignment on each habitat. And so all these are wrong alignments. And so if you do SNP calling, if you look at you do SNP calling individually, you can see, okay, here's a lot of SNPs, these are aligned. And you can see a lot of big difference. But these are all wrong alignments. And similarly here, there's also this 300 base pair insertion. But these are all wrong alignments. If you tackle them separately, you will likely make mistake. And you will see that only 2 reads supported this, and then possibly there's a false positive. But if you jointly consider that you can identify the reads and do the realignment around this region and then recombine it, and you can solve it. You can call the odd SNP and also the long insertions correctly. This is why we need this integration and what's missing in the current tools. And this is roughly our procedure, and basically just doing basically for the face reads and then call the SNP from that and then reface again. And the idea is not new. It's already there. I will also be doing the multiple sequence alignment, local multiple sequence alignment to result. The idea is not new, but we do need better implementation. And overall, that just shows one example. Overall, this is evaluation. And on the old genome in the bottle benchmark, it's actually okay. Here, this on the top, upwards, this shows the false positive number of false positive SNPs, and the SNPs was enough. And below the line, there's a number of false negative calls. You can see this the class 3 and deep variants actually they are doing better than us. This is still sort of early version. We are still improving, but you can see for class 3, for inner colonies is better, mostly because it's doing the phasing. And the deep variant class would have these are doing the phasing, the similar class, they are separate tools, do this variant calling and do phasing and then do variant calling. Okay, and so it is better. But if you look at the more recent, they called Q100 genome in the bottle, the better truth data, the harder truth data. You can see actually we are doing better than the others in terms of both the SNPs and in-house, and also in terms of both of the false positive rates and false negative rates. And so one possibility is actually the older tool might also be overfitting all the data. New data means we are doing select value in the new data, and also the trial variance. And so it's we generally have lower for rates, and the sensitivity is lower than sulfate. This is something we are trying to improve. This is partly caused by the very long deletions. We are currently ignoring that, and partly caused by the random representation, the alignment parameters. But then we are also thinking to integrate the local real assembly. We have local real alignments, multiple sequence alignments. We are integrating the local real assembly. Why is needed? And so this shows the example. This is in a VNTR variable number common time repeats region. You can see there should be two haplotypes. I mean, the numbers are small, but you can just see the insertion positions. They are all over. I mean, it doesn't look like this is deployed. And this is, again, is caused by something like this. It's caused by alignment. And so the better way, actually, for this example, we can resolve with current multiple sequence alignment. And if you do the exam, you can see this, the two haplotypes. And this can also be resolved by alignment because of these events are only several kb, the shorter than rate is fine. But the second example, you can see here the rate coverage reduced. There's a boundary. If you look at alignment, you don't know what's going on. The back to this further example. But this cannot be resolved with this alignment. But if you do the assembly, you can see this caused by this is 100 kilobase deletion in this sample. There's a coordinate, and this can only be resolved with the assembly. Currently, we are developing a local assembler, and we want to integrate this into this long count, this variant color, and to resolve the remaining region. This is one of the reasons why we are not as sensitive as we will be proving. And also, will be this is about doing the. This will talk about a little bit, but I'm not depending this the motivation. Okay, here is we are trying to do the somatic structural variant calling. This is done by joint assembly. And so here in this case, this the red unitics, this is assembly graph. The red ones are tumor only unitics. The blue ones are these are the normal only unitics. We are merging the data, and we know the rename, and we can tell whether what's the source of the unity. And this one, this is mixed, the tumor and not normal mixed. And this is this is schematic graph, and this is a real graph. And we can see here there's a translocation between them. This and it's connected by this by this unity. And so we know there's a translocation. We can use this information to do the translocation counting better than us. And for this, we are this is effectively aligning the long read against the assembly of the normal sample, not the tumor sample. This is not mixed assembly, but back to the normal sample. And then we can see, for example, here the black parts. If you map, okay, the variant color will tell you, for example, a somatic variance, and the mapper will, the color will tell you how many reads are supporting this variant. You can take out the reads and map the read to normal assembly. And if this is real somatic mutation, then the reads should be mapped with the same structural variants. If the fake, then you will discard. And so you can see here, the black part, if you take the reads supporting the structural variant and map to the normal assembly, these basically will disappear, which means that these are very likely to be false positives. And you can see this is actually given variant color, we take the intersection. Generally, if a call is supported by all of them, actually not so many of them are actually false positive. There's a small number of false positive. But for the color only calls, sometimes half of them are false positive, and we can integrate half of them. And in the last part, and in the last adaptation, and we're a bit run out of time. And so this for only we are developing a long range RNA-seq variant called SNP color, and also facing. The important part is actually facing. Variant calling is actually less important. This shows a example. This is a new specific junction we call the ASJ. And you can see this is one haplotype. This is one individual, AC02. We have one individual here, and this the one haplotype, and the other haplotype is facing it here. And you can see this on this haplotype is consistently got spliced, but on the other haplotypes, this is most of them don't have this splicing. And this is mainly caused by this SNP. I mean, you can't really see it. This is caused by a SNP code the first base. In the hydrogen, there's SNP affecting that, and that's affecting the spike signal. And so that's why once there's this SNP and this spreading, efficiency drop a lot. And this one is you can find it in GTEx. And the second one is not in the GTEx. It's like a similar story. This is the red is one habitat, the blue is another habitat. And you can see there's this consistently is missing here. This is the detailed read line. This is very high rate. This shows the subtitle rates. You can see this there's a real specific splicing. And here, this actually is quite far from there, but this is somewhat is causing the splicing difference. And overall, we can do this. Show this example is not in the GTEx. And overall, we can find we can do this volcano plot, half of volcano plot, we call it. And you can see this region with high changes, and also the P value, we can calculate that. And the one interesting part about that is that we know that the human annotation is very complete. We know that almost all the human, below 5% of the junctions, these are normal. Almost all the human splicing are known. But if you look at this ASJ, allele specific junction events, actually more than half, these are actually normal junctions. And the GTEx, their method can only detect junctions that annotated junctions. But our method, when we look at that, actually there are more things that not annotated. These will all be missed by the GTEx. And so there's also a lot of SNPs cannot be explained by nearby SNPs. This could be trans effects. For example, this affecting the ribosome binding proteins and the chain binding, and so it affects splicing. And we don't know but the majority cannot be explained by nearby SNPs. And we also need to double check the number. Actually, I'm not 100% sure these are accurate. We will come back. Anyway, this summary of the third part is the theory of non-recalling is mature, but we need better tool, and we are trying to get something closer to the JDK. There are tools, many tools in development. It's color D, this is for genomic, and the long color is for the RNA sequence. Okay, I will stop here, and so I want to thank my group and also class members and also my collaborators.
Thank you. We already have several questions from the Zoom audience, but if someone here would like to start questioning, Niranjan.
So it was very interesting, the part about error correction really boosting. Is this the classical error correction ideas, or was this something new? Were you incorporating the fact that there are haplotypes where you want to correct based on the haplotype?
Yeah. It's very important that you don't correct away the true SNP between them.
Right.
So the error correction, these are the modern error correction, these are haplotype aware. They try best to only take the read from same habitat and use this read to do error correction, not considering the read from the other. That's critical.
Thanks for the really interesting overview. Do you think there's potential in integrating other data into genome assembly by HiC, something like bioanimal structural mapping or strand-seq?
I think the other technologies, I think this especially like the strand-seq, it's not widely available. It's difficult to produce. Only a few labs can generate data. As for the other, I think the PERC, people have been using the PERC to replace the HiC. I'm not quite sure why it's not widely used. I would think it's they should be fairly similar in terms of for the assembly. These are probably the more popular data types. There are some rarer data types, but like the thing to read, they're also helpful. It's just that they are not widely available, so you will take effort to support this data.
Yeah, so we're talking about a variant called morphosomatic variant. I think most of the examples were for germline slips and so forth. Morphosomatic variant is often low allele frequency. So if you have 40 reads in 2 or 1 in a tumor, or somatic mosaics isn't also on the low frequency. So how do the low reads?
I think the key, they didn't have the slides, and also this hadn't been finished, but we want to do that in the next step. I think the key information is actually the phasing. Because if you have the errors, then you detect the errors that are randomly distributed between the habitats. And if it is a real semantic event, then it should almost all of them should occur to one habitat only. And so you can use this information to distinguish which are the real one and which are the.
So that sounds like it helps you adjust the allele frequency, not so much in the detection, right? Let's say it's only one read out of 40.
One read wouldn't
it's
help.
going to be in one.
Yeah, even two reads, I mean, even two reads, calculate, you can better compare two models. One is
they are ambitious.
Yeah, it's a just by chance that's on the same habitat. But you do still need a few more to support it.
You can only sequence deeper, so next question. Your turn.
Do you see any value for some maybe a new sequencing technology which is very, very accurate, let's say Q60?
It had to be long reads. I mean, if short reads, actually the alignment will be more problematic than the.
Somewhere in the middle, let's say 500 base pair, 600 base pair. What's long enough for you with Q60? That'll give you a benefit, or it has to be kilobases long.
I think it needs to be kilobase long. Otherwise, yeah. I think it's otherwise it will be difficult. It will be used for polishing. I mean, I'm talking about this. Okay, actually, they didn't have that slide. The null has this null polish, and so they do alignment. So they align the read back to the assembly and do the polishing. They can improve the QE by like 3 to 7 points. That actually quite a lot. It means, I mean, if that still hasn't fully used all the information in the reads. And also with this similar approach, I mean, you can use, let's say, very accurate reads. You can fix most of these remaining errors. And so it's fully useful for this.
We had such technique like error-free sequencing technology. Could we replace the kind of approximative overlap or mapping algorithms with some fast, exact Fourier matching or something like that?
I think there are still errors. So, and also when doing the assembly, actually the other habit actually some not often, but sometimes it helps. It helps you want to know there's another habit around it. This will tell you the information about, say, the coverage, whether this is a default region or this is a repetitive region. Not only the rate comes from same habit. So you will still want to see the rates from the other habit. Just don't want to do the assembly. And in habit, this is actually quite important.
We have one interesting question from some audience. What is your recommended error correction method for high-fi reads? And I have additional question. Is it more to try to even more improve high-fi reads now?
I think the error correction, I think there's still room for improvement. The high-fi error correction. Probably the room not a lot, and also there are actually no separate high-fi error correction tools. And although you can run, say, run Hero, you can run this error correction type separately, and similarly for high-fi, you can run the error correction type separately. But however, the following algorithm is actually it's not only the error correction. I mean, need to work with the following steps, and then the error correction still have errors, and the following step need to be aware of what type of errors. I mean, the following step have. And if you feed the Hafezan corrected reads to work code, it's probably not going to work and vice versa.
Okay, thank you. So, another question. So, do Hafezan support pure ultra-long data assembly?
Sorry, what's the question?
So, pure ultra-long data assembly.
Okay,
pure. So, using only ultra-long reads.
Yeah, this is one limitation with the Hafezan is I can talk about that. It's they would say the curse of the overlap assembler. Is that if you have container reads, if one read contains another, and you would think that if container is no information, you should discard container reads, but it's actually not like that. If it's some small fraction of container reads carries information, if you throw away the container reads, and it will create a assembly break, and this is fundamental problem with the overlap-based assembly, and so far we don't have solutions. And with the ultra-long reads, because their read length can span from the 10 kilobase to 1 megabase, and so this containment will happen a lot more often. Although HyperLM doesn't work so well, it has theoretically to try to alleviate that, but not really solving the root problem. And so HyperLM can work, but it doesn't work so that well. Workflow is much better at doing this offline only assembly. But I think actually the workflow team has tried quite a lot of this. And they have also tried to use HyperLM to correct the offline rates and feed the rate to workflow. And they say the comparable assembly to Hero plus workflow assembly. And also, but the problem is often we don't get the offline. It's difficult to get offline rates. And so that's why our group are not focusing on the offline only assembly, more because of especially for other species, it's very difficult to get offline rates.
Just another question. It is similar to Andrew's question. How to address issues caused by tumor heterogeneity and tumor purity in assembly of tumor genes?
I don't have solutions. That's why it's even quite a lot of many work with the chat the correct, and we really don't have good solutions.
In the same vein of thought, right? Nanopore reads actually have more information than just the base content, right? They can give you information on methylation things, right? So perhaps if you have a tumor sample which has cells from different sort of cell types, and some of these rare variants are coming from a particular cell type, maybe you can exploit, you can go beyond the sequence.
Yeah, that's a good point. That's a good point. Yeah. Actually, I was recently talking to my colleagues. He's actually interested in the mosaic implementations, and he said that I haven't studied this, but he told me that the methylation is actually more that the mosaic methylation is actually more frequent than say mosaic SNPs, and it's actually this you can use this information to separate the clones, subclones, and improve. I haven't tried, but good suggestion. Yes.
Next questions.
Yeah. So actually, I'm interested in the tumor somatic calling, and I'm actually wondering whether, for example, if you want to call variants in really complex events, do you think a really high quality reference of the region would improve that? And then is that a good strategy to look at subclonality, for example?
I think for complex region, you really want to do it. I think you should do. You can get tumor non-normal pairs. I think from only tumor sample, it will be very difficult to distinguish which are the difference between the samples. I mean, between the germline samples versus this is also many events. But once you have the good normal assembly, this will make things simpler. And I didn't show here. Where is it? Okay, this one. There's actually a very this is the interesting case. This is if there's I think 6 to 7. Where is it? I think. Anyway, I think that there's a 6 to 7. Okay, it's not here because it's false. There's an inversion on the chromosome 7, and that region actually it's a cyclic duplications. It's a very common region. It's somatic immersion in a very common region. And so if you just do the reference-based alignments, and so it's and this region is similar to chromosome 6. And so if you do reference-based alignment, you will see a translocation with very strong support from the DKTs. But actually, this is a false signal because because the two germline and somatic event entangled together, you cannot separate them out, and this causes the issue. But if you align the read to the personal assembly, and you can clearly see that's the inversion, but you don't see any translocation, no reads according to the translocations. And so this the normal assembly is very helpful for these common events.
Thank you.
Another question from Zoom. What are the main challenges and problems that sequence alignment or genome alignment currently faces and needs to solve?
I think the read alignment, I think this actually one simple. That's a very simple problem, but I think someone need to do that, which is the align the reads back to its own deploy assembly. So far, there are no good tools for that. I mean, you would think this is an easy problem, but it's actually a hard problem. The problem is easy. You just need a specialized implementation for that. The problem with minimal tool is it's only used up to 28 words, but it's not long enough. For examiners, they use 51 home compressed keywords. You need a very long keyword to separate these centimeter copies, and you need a bit specialized alignment for that. And for the genome alignment, that's a whole other topic. I think this people don't know what is the good, what is the right alignment. And say that the TYP2T genome and the align Z02, people don't know what's the good alignment. What's the correct alignment? People don't know. It's a lot harder.
Sorry, keep asking questions. One would say a lot of the developments in the field, the improvements in the field, were driven by really great advances in sequencing technology and the bioinformatics catching up to it, being able to exploit and leverage that. But what do you think is still maybe a more purely computational bottleneck, right? Do you think there is a computational bottleneck that still leads, that if you solve it, it'll be a big jump, independent of the sequencing technology?
Honestly, I don't see there's but depend on the impacts. Because I don't see there's something this very high impact. I don't see that. I think so far, if you look at history, it's still mostly driven by the sequencing technology, short reads, long reads, accurate long reads. I think this the breakthrough is driven by technology.
Is there a breakthrough efficiency that we can imagine? Like, could you imagine IFSM getting 10 times faster or 100 times faster?
I think a few fold faster is still possible, but it's maybe not 10 times or more. And in practice, it's also, I mean, even in the short read days, we were thinking this basic biology courses, it's when you do sequencing, you need even 5. I mean, the actual cost is still more than $1,000 if you count the instruments, capital cost and human cost. It's still more than $1,000. And so if you improve the number, it's a nice. I mean, the cost reduction is much smaller for that. So it's mostly should we focus on this or use your time on something else? It's more balanced. For us, I mean, I try to see same problem in kind of different way.
We keep talking about assembly being a computationally expensive, but the longer the reads get, it gets easier in some sense. And if you can really parallelize it, for example, the assembly task, and there was a lot of work in the early days about how to parallelize assembly, right? We sort of get forgotten that in some sense. But if you bring that back and you make assembly of a human genome not a whole day's operation, but 10-minute operation, then people will say, well, why am I calling variants with mapping and so on? Just first create the reference and compare references to references, and just purely work based on that.
I think there's quite a long way to that. First, the performance, and secondly, to make the example reach that's as good as I said, it is worse than long read for small variants. If you bring that to that level, it's very difficult. It's just assembly is intrinsically harder than the read alignment. Kind of make more mistakes and during this process. I mean, that still can be improved, but I don't know how much. I think it can be solved. The information is there. You just need to take efforts. And then the question back is, do you spend time to solve this or spend time, say, to do the joint assembly and ONT?
On another question from Zoom, from structure and comping variance calling, what is the recommended read and config N50 for long read sequencing?
I would say it is for human, it's typically just one flow cell. I mean, you just do whatever with this one flow cell. I would say you don't need to think too much about that. But if you look at longer reads, you get lower base quality. And so this actually, and also the improvement is not that clear. If you change, then you do see something, but I would say rather small. But generally, I would say no need to think much, just follow.
Okay. Any other questions? Zoom questions? I think I asked most of them. So I have one which is maybe more for nanopore, but the question is, why did you say that anti-duplex is failed? I can still see it on their website. So I don't know if you are perfect to answer this question.
Yeah,
Maybe we need some nanopore.
and now, I mean, I didn't first hear this from nanopore. I mean, it's from they used to work with nanopore. And now I definitely not from my past colleague. I think it, but actually, okay. But actually, I would refer that it's a the duplex is there to compete with high-five. I don't know because you couldn't do at that time. You couldn't do the assembly with similar rates, and now with Workle and especially actually, I believe. Actually, again, I don't have internal information, but I believe they actually stopped developing the duplex when you have the hero already. It's if you can do the assembly with the similar rates, then why is that effort? It's a additional engineering effort to make the duplex. And basically, the throughput at least halved the duplex situation. And so I think it's kind of similar to the time question. And when you have this company have limited efforts, and I think it's better just to make this solids, make similar solids, and possibly make this parameter tool with this parameter tool. Actually, I'm very interested in that, but I don't know what's the practical problem with this. Why it's still.
Yeah, no, I was thinking about Niranjan's question, your discussion about like whether we can do assembly in 10 minutes. And I think it sounds like, the assembly problem, just like this. And, in computer science, we'd like to solve this using just data and like without you looking at other genomes and we just want the reads and then, efficient computational methods to solve that problem, which is great, but then you need a lot of DNA, right? You need to sequence potentially all that. But imagine if you have like the genomes of 1 million individuals and you had stored that in some data structure. Do you think you'd be able to solve the problem quicker? Like, having access to all the data from all these individuals, would you need less data and could you then solve the problem quicker?
Yeah,
I don't know.
We try that. I mean, this also is kind of field project because the developer has left. And we did try to use the cell storage, but the bacteria, I mean, we haven't solved a huge amount. But we take the bacteria long read assembly, the complete the T2 here, they don't have T telomeres. But anyway, it's the complete bacterial genomes. We have hundreds of them. And we want to use that to guide the short rate assembly. I would say this there's some limited success. I think there are two information in it. But it's also kind of tricky. Why it's different? But if you want to recognize the germline genome, it's actually, I mean, there's a common say this the common variant, the common germline genome. I think that should be doable. But when you do this the genome, you actually are into this in private, this basically very rare little specific weights. And so there's some chance that you may miss that. And so I think this is worth exploring. We did very, very early things, but I think that's still room for improvement. But just when you define that, be careful about the biological questions you want to answer with this.
Okay, thank you. Thank you, Heng. It was really. Just one thing, I think Eliza wants to make a photo of us or.